Okay, good evening. Hello. We're going to look at a little video now, just hoping to talk you through the stages involved in mitosis, the reason why it's important, and a couple of maths questions that have been associated with this topic. Okay, some similar key terms that you need to be aware of before we start looking at mitosis. There are 5 of them I just want to draw your attention to. The chromosome. First one. That's that thread-like structure made of protein and DNA, and that's what contains the hereditary material that gets passed on from one generation to the next. During the cell cycle, that genetic information is replicated, and once it's replicated, and as those two copies are joined to one another, they are said to be chromatids. Now, these two chromatids are joined by a centromere. Hopefully this will make sense as we go on to the next few stages. Now, during mitosis, there are 2 important organelles called centrioles. Now, they play a really important role in separating the chromatids and breaking the centromere, and they're found in the nucleus, and they're often referred to as centrioles and being found in the poles of the cell. Before the DNA condenses into chromosomes and it's in its what's a diffuse form, this is the vast majority of the cell cycle. It's known and referred to as chromatin. Okay, so very similar words, but very different meanings. Chromosomes, chromatids, centromere, centriole, and chromatin. Okay, so the importance of mitosis, why it's required. Remember, mitosis creates 2 identical cells. Growth is the most important thing. It ensures that all the cells that are produced by an organism have got the same genetic information from that original zygote, from that original fertilised egg. With that DNA that's in there, mitosis creates identical cells and ensures that they've all got exactly the same DNA. Once differentiation has occurred, so during development, mitosis creates identical cells, ensures that those cells that have differentiated, when they divide and reproduce, you end up with 2 identical copies of those differentiated cells, and the same for repair as well. Obviously, if you want to repair damaged or dead cells with new cells, you need to make sure that they've got exactly the same structure. So mitosis providing 2 identical copies of the same cell ensures that happens. So I thought I'd just run you through this. These are all the stages, and then we'll look at them in a bit more detail in the next few slides. So interphase, this is the first stage here, and if you looked at the previous video, it talks about interphase. That's when the DNA replicates. Chromosomes are invisible, they're in that form, they're in that chromatin form, and it's just the cell readying itself for the division. The first stage of mitosis is prophase. What you've got here is the chromatin is now in a condensed form, and chromosomes are now visible. It's been replicated, so each chromosome is made up of 2 chromatids joined at a centromere. What happens is the centrioles here and here release spindle fibres, and they attach to the centromeres of these chromosomes, and they align, and you can see them in metaphase now, this is the next phase here, they align them along the equator here, so they're all aligned. These spindle fibres then condense and contract, and they pull and they break the centromeres, and we're now into a phase called anaphase, and this is where the chromatids, so they're not really called chromatids, are now broken, and they are now being pulled towards those centrioles. Telophase is when the chromosomes have now reached the centrioles. They now revert back to their diffuse form, long and elongated, and the cell is just about to undertake something called cytokinesis. That's when the cell actually splits in 2. So you've got the 4 phases: prophase, metaphase, anaphase, and telophase. One way you could remember it is, please make another 2. Prophase, metaphase, anaphase, telophase. So we'll have a look at these stages in a bit more detail now. Before we do that though, one thing that the exam board do state is you must be able to recognise the stages of mitosis from the drawings, which we just saw on the previous slide, but also from photographs as well. So hopefully these next slides will run us through that. Here's late interphase, so this is after the DNA is replicated. In both diagrams here, the drawn one and the photograph, here's the nucleus just in here. You can't see the individual chromosomes. The DNA is here in this diffuse chromatin form. We're now ready to enter mitosis itself, the first stage of the mitotic cell division. So prophase. Prophase, you can see, a little bit different now to before. You can actually start to see these distinct chromosomes. There's no structure to them at all. Here's 2 centrioles, right there and there. One of them will migrate around to the other pole of the nucleus, and you can see them here. In this case we've only got 2 pairs of chromosomes here. You can see the centriole there, but they're not lined up in any way. Once we go into metaphase, however, these are now aligned along the equator. You can see the chromosomes aligned along the equator. The centrioles have released these spindle fibres and caused them to align. On the photograph here, here's the centrioles here and here. Spindle fibres in green have been formed, and it's caused the chromosomes to align along the equator. Once we're into anaphase, the centromeres have split. It's caused the 2 chromatids to separate, and they're now called chromosomes again. And you can see there's now a gap forming here as the chromosomes are being pulled back towards these 2 poles, these 2 centrioles. We're now on telophase. You can't really see them being particularly as chromosomes. Revert back to their more diffuse chromatin form. Spindle fibres are pushing them apart again as well, and we're now starting to enter cytokinesis. This cell's going to split into 2 different cells. Here's another diagram. Here's interphase. You can't really tell or identify the chromosomes. Prophase, the DNA, the chromatin has now formed its more condensed form, and you can see those chromosomes, but they're not showing any alignment or any structure. Metaphase would look a bit like this. I draw cell membrane, draw the nucleus, can draw a centriole here, a centriole here. What you'd end up with are the chromosomes aligned across the equator, and each join there with the centriole. Sorry, with the centromere here, and here, and here, and here. Here's the centrioles. Anaphase, the spindle fibres have pulled in this direction and that direction, separated the centromeres, split the centromeres, so the chromosomes are now heading back towards the centrioles. And telophase now, they've now reached the centrioles, and cytokinesis is beginning to occur here. Okay. Here's a few stages we can try and identify. Well, E here, this one's got to be interphase, because it's the DNA, the chromosomes, you can't identify them as chromosomes, it's in that condensed form. I would probably then go to say this has got to be prophase. You can identify them as being chromosomes, but they're not in any structure, they're not aligned in any way. C must be metaphase, they're now aligned along the equator. B must be anaphase, they're now being pulled towards the centrioles, towards the poles. And telophase here, they're now forming, we're almost ready for 2 distinct nuclei, and you can just imagine there's going to be a split just running down here in a second, cytokinesis. Here's a few more stages. This is an example of what you might look at down a microscope. And if you just have a look around, this is probably a good example of one in Interphase. Here's one in Prophase. You can see them as chromosomes, but they're not in any structure in any way. This is probably a good one in Metaphase. You can see them as chromosomes, and they're aligned in some fashion along the equator. Here's anaphase that just started to be pulled apart, and this is getting towards telophase up here. Last questions that you might get asked. Here's some data. Somebody's been looking at maybe a root tip squash, looking at some cells and seeing the number of cells at each stage. And you can see mitosis, interphase making up the vast majority. Prophase 73, 20, 9 and 8 for the relative stages of mitosis. If one cycle, so one cell division, takes 20 hours in this organism or whatever we're looking at here, how many minutes would metaphase take? Now the important thing is, and it always gets stated, you've got to assume the number of cells at each stage, i.e. here, is proportional to the length of each stage. I'll ask you to show you working. So the first thing you need to do is add up all the cells, see how many cells you're dealing with. So 890 add 73 add 20 add 9 and 8 equals 1,000. So we've got 1,000 cells in total. We're only interested in metaphase, so that's 20 divided by 1,000. So that gives us 0.02. So 0.02 of the cells are in metaphase. So we've been told that the cell cycle lasts 20 hours. So 20 hours times by 0.02 equals 0.4 hours. 0.4 hours is okay. If I want to convert it to minutes, 60 minutes in an hour obviously, times that by 0.4, so that gets you your 24 minutes. There's one question you might get asked. Another question you might get asked is, in what percentage of the cells would the chromosome be invisible? That's assuming those numbers before.